Cellular respiration is a very complicated process that helps create an energy carrier called ATP, which is a complex organic chemical. Every living organism uses ATP as a way to metabolize and store energy. Through cellular respiration, we are able to create enough ATP to power our bodies, and it consists of four processes. This includes glycolysis, link reaction, Krebs cycle, and the electron transfer system, where most of the ATP is made. Glycolysis begins the progressive breakdown of glucose and occurs in the cell cytoplasm. First, glucose is activated by two molecules of 2-ATP. It is then split into two 3-carbon molecules, each of which is oxidized, loses a hydrogen pair, releases 2-ATP, and leaving 3-carbon pyruvate or PGA. This completes glycolysis. In glycolysis then, glucose is split in two, and a net total of 2 ATP are produced for each glucose molecule metabolized. Glycolysis is followed by a link reaction, where pyruvate loses a hydrogen pair to the acceptor molecule NAD+, to become NADH, which moves to the electron transfer chain. Pyruvate also loses CO2 molecule and combines with coenzyme A to become 2-acetyl-CoA. Acetyl-CoA then leaves the cytoplasm and enters the Krebs cycle in the matrix of the mitochondria. The purpose of the Krebs cycle is to progressively strip off more high-energy hydrogen pairs from the glucose molecules and send them to NAD+, and then the electron transfer chain where they will be used to make ATP. The Krebs cycle begins when 2-carbon acetyl-CoA joins in with the 4C molecule that is finishing the cycle to form 6-carbon citric acid. This molecule is rich in high-energy hydrogen pairs. The rest of the cycle will be devoted to the progressive oxidation as hydrogen pairs are stripped off from molecules and accepted by NAD+ to become NADH, which will move to the electron transfer chain. CO2 molecules are also removed during the Krebs cycle, leaving behind 5C and then 4C molecules. The removed CO2 is released as waste product by diffusion and exhalation. At the end of the Krebs cycle, the remaining 4-carbon joins with another 2-carbon acetyl-CoA, forming 6-carbon citric acid. The cycle continues as long as there is adequate amount of glucose undergoing glycolysis. High-energy NADH molecules have been produced in glycolysis. In addition to the Krebs cycle, when NAD+ and FAD+ is reduced by hydrogen atoms. In the final step of aerobic cellular respiration, NADH releases an H+ ion into the mitochondrial matrix, and two high-energy electrons enter the electron transfer chain. Each chain is made up of 4 complexes that lie embedded in the inner mitochondrial membrane. When NADH molecules enter the first complex, protons, H+, and high-energy electrons are split away, leaving NAD+. The high-energy electrons are passed down the chain from one complex to the next. The accumulation of H+ sets up an electrochemical gradient between the inner membrane and the mitochondrial matrix. The potential energy in this gradient will be used to drive the production of ATP during chemiosmosis. When 2 electrons emerge from the last complex in the ED chain depleted of energy, they join 2 H+ ions that will be released from ATP synthase. After chemiosmosis, oxygen and the reactant for cell respiration combines with the present electrons and H ions, forming H2O as a waste product.